Hello everyone. In this presentation, we'll be studying about enzyme inhibition. Now, enzyme inhibition are of mainly two types, irreversible inhibition and reversible inhibition. As name suggests, irreversible inhibition is the inhibition which cannot be reversed back. And reversible inhibition is the inhibition which can be reversed back. Okay, now reversible inhibition are of mainly three types, competitive inhibition, non-competitive inhibition and uncompetitive inhibition. In this presentation, we'll be studying about all these types of inhibition. So, enzyme inhibition are of mainly two types. We saw, no? Irreversible inhibition and reversible inhibition. So, first we'll be studying irreversible inhibition. As the name suggests, irreversible inhibitions are the inhibitions which cannot be reversed back. Now, what does irreversible inhibitors do? Now, irreversible inhibitors usually bound covalently to the enzyme and destroys the functional group in the active site. So, once your functional group in your active site is being destroyed, the enzyme becomes inactive. And once the enzyme has become inactive, it cannot do its catalytic activity. So, what is the function of irreversible inhibitors? The irreversible inhibitors destroys the functional group present in your active sites, and therefore once your functional group in the active site is being destroyed, the enzyme becomes inactive. Now, most of the irreversible inhibitors are toxic substances. Now, with this diagram, let's understand how your irreversible inhibitor inhibits your enzyme activity. Okay, this is the enzyme and this is the substrate and this is the irreversible inhibitor. This irreversible inhibitor covalently binds to the active site of the enzyme and destroys the functional group present in your active site. And once it destroys the functional group present in your active site, the enzyme becomes inactive and it cannot do its catalytic function properly. Okay, and your substrate also cannot bind to this enzyme. Okay. Now, the examples of your irreversible inhibitor is your cyanide. Now, this cyanide usually inhibits your complex number 4 involved in your respiratory chain enzymes. That is your enzymes involved in your electron transport chain. Now, cyanide is acting as an irreversible inhibitor. It binds to the copper present in your complex number 4 of your electron transport chain. Second example is diisopropyl phosphofluorate, which inhibits your enzyme acetylcholine esterase. Now, this diisopropyl phosphofluorate also inhibits other enzymes which has serine in their active site. Now, coming to reversible inhibition. As the name suggests, reversible inhibition, the inhibition which can be reversed back. Okay. Now, reversible inhibition, and the inhibitor can dissociate from the enzyme. Now, that means the binding between the inhibitor and the enzyme is not that strong. Okay. So, reversible inhibitors are usually binding non-covalently with the enzyme. Okay. And there are mainly three types of reversible inhibition: competitive inhibition, uncompetitive inhibition and non-competitive inhibition. Now, this diagram is helping us to understand how your inhibitors are binding to the enzyme. In competitive inhibition, the competitive inhibitor binds to the active site of your enzyme. Okay. The non-competitive inhibitor usually binds to the other side, not in the active site, other side. You call it allosteric site of the enzyme. An uncompetitive inhibitor usually binds to the enzyme substrate complex. Okay. Now let's understand what is competitive inhibition. The competitive inhibitor structure is very much similar to that of substrate. Okay, can you see the structure of your substrate and the structure of your competitive inhibitor is same. The competitive inhibitor binds to the active site of your enzyme. Okay, and it usually binds reversibly to the active site of your enzyme. See, when the enzyme binds to your inhibitor, it becomes inactive. Okay. Now, this reaction can be reversed back. That is, your inhibitor can get dissociates from the enzyme, and the reaction can be reversed back. Now, when your substrate binds to the enzyme, it forms an enzyme substrate complex, and the enzyme acts on your substrate and converts the substrate into product. Now let's understand this. This is your enzyme and this is your substrate. The enzyme is acting upon your substrate to form enzyme substrate complex, and this enzyme substrate complex is later being broken down to enzyme and product. Okay. The formation of your enzyme substrate complex is a reversible reaction. Okay. to the enzyme, to the active site of your enzyme. Your inhibitor or competitive inhibitor also binds to the active site of your enzyme to form your enzyme inhibitor complex. Now the structure of your enzyme inhibitor complex and the structure of your enzyme substrate complex is very much similar to each other. Now here the activity of your competitive inhibitor can be inhibited by increasing the substrate concentration. Why? Because we know that the substrate is also binding to active site of your enzyme, and this competitive inhibitor is also binding to the active site of your enzyme. Okay. Now, when you increase the concentration of your substrate, then the chances of substrate binding to the active site of the enzyme will be more. Okay. So, the reaction will proceed in this direction, leading to the formation of enzyme substrate complex, which can be broken down to enzyme and product. Okay. So, you can inhibit the competitive inhibitor activity by increasing the concentration of your substrate, because the competitive inhibitor and the substrate is binding to the active site of your enzyme. Both are binding to the active site of your enzyme. A classical example of competitive inhibition is your methanol poisoning. The methanol is being converted to formaldehyde by enzyme alcoholic dehydrogenase present in our body. Okay, now the formaldehyde is highly toxic to the body. Okay, so what is doctor doing? He is giving you high doses of ethanol. The ethanol is binding to the active site of your alcoholic dehydrogenase, so as and inhibiting the activity of alcoholic dehydrogenase. In other words, ethanol is acting as an competitive inhibitor for your enzyme alcoholic dehydrogenase and inhibiting the activity of your alcoholic dehydrogenase. So what is doctor doing? He is giving you high concentration of your ethanol to prevent your methanol poisoning. So here this is a Michael-Menten's graph for competitive inhibited one and uninhibited one, and this is a Lineweaver plot for the competitive inhibited and uninhibited one. So from these graphs you can see that the Vmax remains same for competitive inhibited and uninhibited one. Can you see the Vmax remains the same for competitive inhibited and uninhibited one, and the Km but increases. So during your competitive inhibition, the Km value increases. Can you see here? The Km value increases but the Vmax remains the same. So during your competitive inhibition, what is happening? The Km is increasing but the Vmax remains same for the uninhibited and competitive inhibited one. Okay. So you have to remember during your competitive inhibition, what is happening is that Vmax remains a constant but Km increases. This is the one which this point which you have to remember, which will help us to solve other problems. Now let's understand what is happening during your non-competitive inhibition. Now during your non-competitive inhibition, the non-competitive inhibitor is binding to the enzyme on the allosteric side. Okay, so now here the inhibitor is binding not to the active site but to the allosteric site of your enzyme, leading to a conformational change in structure of your enzyme, leading to so that your substrate cannot bind to the active site of your enzyme and that the reaction cannot take place. Okay, so what is happening here is that the inhibitor is binding to the allosteric site of your enzyme, leading to a conformational change in structure of your enzyme. Can you see here the active site was your triangle in the triangle form. Once your inhibitor has bound, there is a change in structure of your active site. Now to this active site your substrate cannot bind and there is no reaction. Okay. So here the inhibitor is binding to the allosteric site of your enzyme, leading to a conformational change in structure of your enzyme so that your substrate cannot bind to the active site and there is no reaction. The non-competitive inhibitor is binding to the allosteric site of the enzyme, leading to the conformational change in structure of the enzyme so that the enzyme cannot catalyze the reaction. Okay. So the non-competitive inhibitor is not binding to the active site but instead it is binding to the allosteric site of the enzyme, leading to a conformational change in structure of the enzyme so that the enzyme cannot catalyze the reaction. The non-competitive inhibitor binds to the free enzyme as well as to the enzyme substrate complex. And the non-competitive inhibitor's activity cannot be reversed by increasing the substrate concentration. Okay, here in non-competitive inhibitor is binding to the allosteric site of the enzyme, leading to a conformational change in structure of the enzyme so that the enzyme cannot bind to the substrate and the reaction cannot take place. Okay, so this is your Michael-Menton's graph for the non-competitive inhibited and uninhibited one, and this is a line-weaver plot for the non-competitive inhibited and uninhibited one. So from these graphs, you can see that the Vmax decreases during the non-competitive inhibition, but the Km remains constant. Okay, so from these graphs, you can see that the Vmax decreases during your non-competitive inhibition, but the Km remains constant. Okay, so during your non-competitive inhibition, the Vmax decreases, but the Km remains constant. Okay, so you have to remember that during your non-competitive inhibition, Vmax decreases, but the Km remains constant. Now let's understand what is happening during your uncompetitive inhibition. Now the uncompetitive inhibitor binds to the site distinct from their active site. Okay, and the uncompetitive inhibitor only binds to the enzyme substrate complex. We saw that in non-competitive inhibition, the non-competitive inhibitor binds to a free enzyme as well as to your enzyme substrate complex. But your non-competitive inhibitor binds only to your enzyme substrate complex, and it binds to the site distinct from your active site. Now this diagram is showing how is your uncompetitive inhibitor acting. Now uncompetitive inhibitor only binds to enzyme substrate complex. Okay, so can you see red color is the uncompetitive inhibitor which is only binding to enzyme substrate complex, leading to the inactivation of your enzyme so that the enzyme doesn't catalyze the reaction. Okay. The uncompetitive inhibitor binds to the allosteric site of the enzyme and it only binds to the enzyme substrate complex. The uncompetitive inhibitor is structurally not similar to your substrate. Okay, so can you see the uncompetitive inhibitor is only binding to enzyme substrate complex, leading to the formation of enzyme substrate inhibitor complex, and Ki is the rate constant for this equation, that is enzyme substrate complex plus inhibitor is giving rise to enzyme substrate inhibitor complex, and Ki is the rate constant for this equation. So Ki is equal to a concentration of enzyme substrate complex into concentration of inhibitor divided by concentration of enzyme substrate inhibitor complex. Okay. So uncompetitive inhibition. So this is a Michael-Menton's graph for uncompetitive inhibited and uninhibited one. And this is a line-weaver plot for uncompetitive inhibited and uninhibited one. So from these graphs, you can see that during your uncompetitive inhibition, Vmax and Km both decreases. Okay. So you have to remember that during your uncompetitive inhibition, Vmax and Km both decreases. Now this slide is showing comparative study of your competitive inhibition, uncompetitive inhibition and non-competitive inhibition. Now during your competitive inhibition, the competitive inhibitor binds to the active site of your enzyme, leading to the inactivation of your enzyme. The structure of your competitive inhibitor is very much similar to the structure of your substrate. Okay. During your competitive inhibition, Km is increased and Vmax remains constant. During your non-competitive inhibition, the non-competitive inhibitor binds to the allosteric site of your enzyme, okay, and it leading to the conformational change in structure of your enzyme so that the enzyme cannot bind to your substrate and your reaction cannot take place. Okay, during the non-competitive inhibitor binds to your free enzyme as well as to your enzyme substrate complex. During your non-competitive inhibition, Km remains constant but Vmax is reduced. In your uncompetitive inhibition, The uncompetitive inhibitor binds only to your enzyme substrate complex, leading to the inactivation of your enzyme. Okay. During your uncompetitive inhibition, Vmax and Km both are reduced. Okay. So this is a Lineweaver plot for your Lineweaver plot for your competitive inhibition, uncompetitive inhibition and non-competitive inhibition. It is just showing a comparative study of your all the three inhibition. Okay. So this slide is only giving you 3 questions, and this 3 question you can definitely answer if you have seen my complete presentation. Okay. Thank you very much for your patience listening.